Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARC Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARC. All statements made regarding companies or securities are strictly beliefs and points of view held by ARC or podcast guests, and are not endorsements or recommendations by ARC to buy, sell, or hold any security. Clients of ARC Investment Management may maintain positions in the securities discussed in this podcast.
Welcome to FYI, the For Your Innovation podcast. Today, we are extremely honored to have Dr. Agnieszka Czechowicz on. So for a quick bio, because we could spend probably the entire time just doing a bio, Agnie is a physician scientist, biotech entrepreneur, 20 years of experience, stem cell biology, translation research. Dr. Czechowicz is also a faculty member in Stanford University's Department of Pediatrics, Hematology, Oncology, Stem Cell Transplantation, and Regenerative Medicine, also a member of Stanford's Institute for Stem Cell Biology and Regenerative Medicine. Dr. Czechowicz completed her PhD work in developmental biology at Stanford with Professor Irv Weissman. She then completed her residency in pediatrics at the Boston Children's Hospital and then pursued subspecialty training in pediatric hematology and oncology at the Dana-Farber Cancer Institute while simultaneously conducting postdoctoral research with Professor Derek Rossi in collaboration with Dr. David Scadden, and got her MD and PhD and bachelor's degree from Stanford. So lifer for Stanford. But that was all incredible. And welcome to the podcast. Thanks for joining us. So let's dig in. There's so much to say, but first just a welcome.
Thank you so much for having me. I really appreciate it. It's lovely to get a chance to speak with you today.
Of course. So let's start with your clinical and research interests. So a bit of the how did we get here after hearing your incredible bio. So maybe what would be helpful if you could just describe your sort of path to success within Stanford and the transplant medicine ecosystem. Was this something you kind of always knew you wanted to do or thought you would always do, or did it kind of surprise you?
I guess it's a little bit of both. So I fell in love with transplant medicine at a very early age. I actually had a distant family member who was diagnosed with a blood cancer and was offered a bone marrow transplant many, many years ago when I was growing up. And that first really inspired me to learn more about stem cell transplantation and stem cell medicine. And I started to do research in the area back as a high school student at the University of Minnesota, and then really bit the bug. And then as you heard from my bio, I have had a longstanding history at Stanford and first showed up on the farm as an undergrad and had the great privilege of training with Irv Weissman, who is one of the great stem cell scientists and pioneers in the field. And he further inspired me to continue studying stem cells and thinking about how you can apply them for the curative treatment of so many different diseases. And so in some ways, I have been in this area for an incredibly long time, over 2 decades now. But in other ways, it still continues to surprise me that I'm still in this area, that I found it so early, and that I continue to learn new things about it on a daily basis and continue to think that we're just at the tip of the iceberg in understanding the system and applying it to so many different disease processes. So it's been a wonderful journey so far, but still lots of work ahead of us.
That's awesome. I guess hearing what you're saying, it sounds like there would have been a lot of challenges and maybe a lot of interesting tidbits along the way. So just wondering if you can share with us maybe some of the not so great parts of it and maybe some of the better ones too.
Yeah, so I think the road to becoming a physician scientist is just a long one. There's many different parts of the process. In part, that's because both pieces of it, being a physician and being a scientist, take a lot of learning and a lot of depth to really understand those areas and be able to contribute fully. And when you do both, and you kind of have 1 + 1 that you also have to really dive deep on. And so just the path itself has been a longer one, given the amount of knowledge that one has had to learn and wanted to learn. And Every part of that process has its own unique components to the journey, everything from graduate school to residencies to fellowships to being a junior faculty member. And so there definitely have been highs and lows on this journey, but I've been incredibly inspired by our patients and by our science. And those are definitely the highlights. While there are some patients who unfortunately really don't do well and some experiments that don't turn out, on the flip side, there are many moments where you're really motivated to do better and to see the impact of the treatments that we have available. And then similarly, every now and then we have some extremely exciting research results that really motivate us to continue to work in this area and give us really strong reason to believe that we can transform this field in powerful ways to then influence the patients that we see. And so I think it's been a long journey, still at the very beginning of this journey in many ways, lots of more work ahead of us with some bumps along the way, but also a lot of inspiration, a lot of amazing outcomes for our patients with these types of transplant procedures, and then a lot of amazing data that we've generated, especially in the non-genotoxic conditioning space, where we've been able to show that we can completely flip out a blood and immune system without any toxicity in animal models.
We will certainly get into that exciting data that you have been a huge contributor and part of. But maybe just to take it back a little bit, to give context kind of into your clinical interests. So, your clinical interests are extremely vast, but among them are bone marrow failure, and especially Fanconi anemia and aplastic anemia. So first of all, for anyone on the call who maybe doesn't know what Fanconi anemia or aplastic anemia are, it would be helpful maybe if you could give some context there, and maybe just why you became so fascinated with these types of bone marrow failures.
Yeah, absolutely. So my interest in these diseases really start from a higher level interest in the hematopoietic system and in the power of hematopoietic stem cells. So these are these very unique cells that can be found in the bone marrow or can be pushed out of the bone marrow into the peripheral blood that have this amazing capacity to be able to self-renew for life and also give rise to the entire blood and immune system. And as many of you know, the blood and immune system has so many different reasons that it's important. And as we continue to learn about it, we continue to learn more and more ways that it is impactful in the generation of macrophages and the generation of microglia. It's interaction with basically every component of our body from the very beginnings of life through the aging process. And the blood and immune system can go wrong in many different ways, and that can cause a lot of different diseases, ranging from genetic diseases to malignant diseases to autoimmune diseases. And now we're learning more and more about interactions with the cardiovascular system, the neurologic system. So it's really a critical system that we really want to maintain in an intact state to preserve health for many different people. And when it goes awry, there's so many different diseases that unfortunately can occur that affect millions of people around the world. And hematopoietic stem cells are these unique cells that can power that entire system. And so through these transplantation processes, we have the ability to replace that blood and immune system with these types of cells, and then can affect all of these disease processes. And while our research group and myself personally study many different aspects of the blood and immune system and its effects in many different diseases, I've started to sub-focus really in these bone marrow failure syndromes as a subgroup of those many different diseases. And in part, these are areas where the hematopoietic stem cells really fall apart and they're not able to maintain that hematopoietic capacity. And so patients unfortunately lose their entire blood and immune system rather than just one aspect of it. And so we've been trying to study that better in various different mouse models and in patient samples to understand how hematopoietic stem cells interact with their microenvironment, how do they preserve that interaction to maintain a healthy blood and immune system, and then where does it go awry in these certain diseases. And Fanconi anemia is one genetic cause of bone marrow failure syndromes that's one of the most common ones. And so that's why we've started to sub-focus in that disease. And it's also a cancer predisposition syndrome. These patients have a over 1,000-fold increased risk of cancer compared to other patients. And so it's a unique place to be able to study the interactions of the blood and immune system, understand the stability of hematopoiesis and where it falls apart, and then also watch the progression of cancer in an accelerated state to then hopefully learn from that and apply it to many other patients with Fanconi anemia, but also many other diseases. And Fanconi anemia, as I mentioned mentioned, is a genetic cause of bone marrow failure that is caused by defects in one of 23 different genes that are involved in the Fanconi complex, which is an important DNA repair complex. And so unfortunately, when patients have mutations in any one of those genes, they lose the ability to repair their DNA, leading to bone marrow failure and this increased risk of cancer. And so I feel for these patients and these families who unfortunately just have had the bad luck to acquire mutations in these genes that then have such profound effects. And it's been a neat opportunity to get to study these patients and their samples, but also really try to help them in profound ways through our standard of care therapies and through our newer treatments that we've been developing. And then aplastic anemia is kind of a parallel to that. It's kind of a catch-all category for patients who have really low blood counts and an empty bone marrow that we don't always understand the causes for. And often it's thought to be probably immune mediated. And there's still a lot to learn about what causes that, what occurs in those situations. And so those two groups of diseases both cause bone marrow failure, but for different reasons. And so give us different opportunities to study genetic causes versus acquired and potentially immune mediated causes.
Yeah, and as you mentioned, sort of the hematopoietic stem cell transplants are so important to these patients, but one challenge to them, as you mentioned as well, is the conditioning that has to happen before. And you've also been incredibly crucial in finding solutions for hematopoietic diseases, including bone marrow failure, because, one example, you're the scientific co-founder for a company called Magenta Therapeutics. So can you kind of give us some background about how you came up with the idea for Magenta? And just by way of a tiny bit of background for Magenta, for anyone who doesn't know, they're developing an antibody drug conjugate. It's essentially for the conditioning for hematopoietic stem cell transplants, amongst other programs. But that's one of the ones we were excited about. And it's designed to really improve the hematopoietic stem cell transplantation.
I'm very excited about what Magenta is doing and think it'll be incredibly impactful for our field. As I mentioned, I got into the area of hematopoietic stem cell research and bone marrow transplantation many decades ago and was really inspired by some of the visions of Professor Irv Weissman, who was my PhD advisor at the time, who had isolated hematopoietic stem cells and showed that those more purified cells could be effective for transplantation with much lower rates of graft versus host disease, which is one of the larger complications associated with transplantation. But one of the problems with purified hematopoietic stem cells was that it was harder to get them to engraft. And so Professor Weissman had given me the challenge of trying to understand why that was. And as a graduate student, had the wonderful opportunity to work with a postdoc, Deepta Bhattacharya and Dan Graft, to try to understand and explore those barriers to hematopoietic stem cell transplantation. And in a series of experiments that we now did many years ago, we really showed that your own stem cells compete for space with the transplanted stem cells in a hematopoietic stem cell transplantation process. And so we explored that level of competition and were really looking at what were those types of barriers. And as part of that, showed that we could eliminate that barrier by depleting one's own host hematopoietic stem cells. And when we did that in mouse models, we showed that if you could deplete them with various different types of antibodies, then you can get engraftment of donor stem cells without the same type of concerns. You could get very high turnover of the hematopoietic system without any toxicity. And excitingly, not just avoidance of toxicity to various different types of organ systems that normally are affected, but also decreased risks of graft versus host disease and other issues. And so that as a concept has been something that we've been studying now for over a decade and have been working to develop various different types of antibody-based conditioning methods that can replace the current genotoxic conditioning that we use today. So right now, before a bone marrow transplant or a hematopoietic cell transplant, patients have to be exposed to total body radiation or genotoxic conditioning, such as chemotherapy agents like busulfan. And unfortunately, these cause pretty significant toxicities to patients up front. They cause profound cytopenias requiring lots of transfusions. They cause all sorts of different types of organ damage. They cause infertility issues, and they cause a delayed immune reconstitution from the new grafts. And with our antibody-based conditioning methods, we've shown that if you just deplete the patient's or mice's own stem cells, then you can get this type of turnover in a completely safe and targeted fashion. And so we initially did these types of studies in mouse models using an antagonistic antibody, and then we further showed that you can make these types of antibodies more potent through antibody drug conjugates. And we had a nice paper that we published a few years ago in collaboration with David Scadden's group while I was a postdoc in Derek Rossi's lab, showing that anti-CD117 antibody drug conjugates could beautifully be used in such a fashion to safely and effectively turn over the entire blood and immune system. And that was really one of the founding pieces of Magenta Therapeutics, was to take that concept that we had shown in mouse models and actually turn it into a therapy for patients. And what I think is really interesting is that although we do lots of these types of transplant procedures around the world, there's actually a recent article that said that as a field, we've done over 1.5 million hematopoietic cell transplants now. All of the agents that we really use today have been borrowed from other areas. There are chemotherapeutics that have been borrowed from treatments of cancer. There are immune suppressants that have been borrowed from organ transplant. And that's why they don't work perfectly for what we do. And I think why the patients have the toxicities that we have, even though, again, these are very curative procedures that can be very effective. And the idea behind Magenta was to really develop therapies directly for hematopoietic stem cell transplantation that are ideal and optimal for what we're trying to do, to maintain that efficacy, but really dramatically change the toxicity profile to make these types of transplants super safe, super easy, outpatient with decreased costs. And so Magenta is on that mission, and one of their programs is around development of antibody drug conjugates, where they've really been working on making the ideal agents from scratch, picking the best antibodies, the best toxins, the best linkers, and have been nicely progressing along that journey. They have reported on some nice data in non-human primates and recently launched a clinical trial in relapsed refractory AML patients testing their CD117 antibody drug conjugate, and recently announced positive news that likely their agent is showing some positive efficacy and safety data, but we'll really see what that looks like in the coming weeks when they start to release more of that.
Yeah, so actually I was going to ask you about that. So yes, this was about 2 weeks ago, I think, that Magenta announced for the 117 program. As you mentioned, it's a phase 1/2 targeted treatment, relapsed refractory acute myeloid leukemia, myelodysplasia as well. We didn't get too many updates on what the data will look like, but I do think we'll see some of that data in the company's earnings call. So any thoughts on what maybe we can expect? I think the company's guided that we may hear things about cell depletion, tolerability, drug clearance, target binding. Any ideas on kind of what expectations should be, or shall we just wait and be patient?
So their study is a dose-finding study and a dose escalation study. And so they have announced that they've treated multiple patients and are on that path to dose escalation. And so I think we'll see how the efficacy overlays where they are in their dosing curves. But from everything that we've seen in the preclinical setting, these agents can be incredibly powerful and incredibly efficacious. In our mouse models, as I mentioned, we've been able to get complete turnover of the entire blood and immune system with one-time doses of proof of concept antibody drug conjugates with really no toxicity. And the results have been so profound that it really leads us to believe this could be game-changing for the field. And excitingly, in our mouse models, we've shown that these types of agents can cause minimal cytopenias, really minimal toxicity to other organ systems, minimal aplasia to the marrow even, and yet, again, complete turnover. And so I think that's what we ultimately are likely to see in patient trials with the antibody drug conjugates that are directed against the human receptors that are coming out of Magenta as well. But I think one of the challenges is that prior to launching into a transplant study, we'll really be able to see those types of effects that are transformative for our field. First, we have to really try to find the appropriate dose and explore this in patients pre-transplant where they can be tested as single agents. And I think Magenta's strategy of doing this in the relapse refractory MDS and AML patients is a very smart one that is allowing them to treat patients more rapidly and test it as a single agent. And based upon what they've said, the fact that it is well tolerated and they're showing positive pharmacodynamic activity, I think is extremely exciting. I think those are the biggest concerns. Is it really going to be as safe as we've seen in our preclinical models? And is it really going to be able to allow depletion of these CD117 positive cells? If the answer to that is yes, then I'm very confident that the rest of the results will be amazing as well. And we actually just had a recent abstract at the TCT conference comparing the different types of anti-KIT agents head-to-head in mouse models and really showed that the antibody drug conjugates were above and beyond all other types of reagents in terms of being able to allow this type of rapid turnover without any toxicity concerns.
So very early, but very exciting and potentially compelling data. And obviously, one of the things I'm really excited about and looking for is that it looks like it was well tolerated, which, as you mentioned, is one of the most important things that we're all looking out for. The company did disclose on about 2 weeks ago as well that they would be halting, other programs in pipeline and maybe kind of pausing some of their earlier target discovery work as well, also potentially reducing the company's workforce by about 14%. That's going to extend their cash runway out to about Q2 2024. Just overall, we're just talking about this, kind of positive data that they seem to have, and we'll hear more probably at the beginning of May. But how should we think about companies' prospects of continuing? It's really difficult market conditions, obviously. And so, the need for this company specifically, but others, to focus away maybe from new target discovery or other programs and needing to reduce staff in order to kind of, keep the lights on. What's kind of your feeling, general market sentiment, I guess?
I think it's unfortunate that the markets are where they are and that we don't have the same type of resources that we're seeing being put into this community that really can use them in very effective ways to transform medicine. I think Magenta is being very smart and strategic about preserving the resources it has to really be able to push forward this incredibly important program. While they are making a number of different types of agents that can be used to improve the transplant process that I think cumulatively will be incredibly important to really completely change the way that we do transplants. I do also think that this one agent is the most important of that batch, and being able to have the resources to really show that and run the trials effectively to then be able to garner more resources to advance the other programs is an important part of that strategy. And so while those decisions are challenging decisions, I applaud them for making them to be able to preserve that capital, to be able to push forward this program. And again, from our preclinical data and the preclinical data that the company has shared, this one agent can truly be transformative. Really, we've seen no toxicity and the ability to completely replace an entire hematopoietic system in our models, which if you think about how we do transplantation today, which is giving patients all these genotoxic agents, having to keep them in the hospital for weeks upon weeks upon weeks, having to give them all of the supportive care, and then seeing these awful complications and the costs associated with this whole process, it really is a horrendous process that, again, we're grateful that we have to be able to offer patients because it really can be transformative for their lives and curative of so many diseases. But when you care for these kids, you appreciate all of the problems associated with it. And if one agent can have such a profound difference in transforming that whole process, I think it is important to give it the resources to really advance appropriately. And then hopefully, again, once they have more data, and more support from the investment community, can restart those other programs, because I think that they are also important, ranging from their mobilization agents to their other target agents as well. And again, applaud Magenta for really being thoughtful about the development of this agent and trying to make the most optimal agent. I know that there were some delays last year as they were changing out one of the components of the CD117 ADC, but I think that although it slowed them down, it will give them a better profile of this agent. And again, the goal was to make the best drug here as that can have the most profound impact on patients. So exciting to see that it's now in some of these patients, exciting to see the data that will come out of it, and then hopefully its ability to be used in many other settings to garner more data and really transform our field.
Awesome. And maybe just for context, we've been talking about hematopoietic stem cell transplantation, but this can also be very important for gene editing. So maybe a minute to speak on gene editing, kind of gene therapy, and how that fits into this whole picture. I think that would be helpful context for people.
As I mentioned, there's a lot of different diseases that are caused by defects in hematopoietic stem cells. And as a community over now many years, we've shown that we can cure these diseases through replacement of hematopoietic stem cells. A majority of the work to date has been using hematopoietic stem cells from healthy donors that don't have genetic defects. But the problem with doing that is twofold. One is that one has to completely immunoablate the host's immune system so they don't immunologically reject the donor cells. And then the other is the potential concern for graft versus host disease, which is this awful immunologic reaction from a donor graft that can affect a patient's entire body, but most profoundly cause pretty significant damage to skin, gut, and liver. And so there's been a big interest in the community to move away from allogeneic donor grafts and instead work on patients' own hematopoietic stem cells through autologous gene manipulation strategies. And so taking patients' own cells, correcting them using a variety of different methods, and then infusing them back into patients. And we've seen a lot of success with these types of approaches using lentiviral therapies, and now are starting to see the advents of other types of gene editing, base editing, and other types of gene manipulation strategies that are very promising. But the issue that still remains is you have to take the stem cells out, correct them, but then you also have to put them back into patients. On the taking the stem cells out, we have some great ways of doing that, although I think we can still do better. And that's where the Magenta 145 compound has the potential ability to better mobilize more potent stem cells. But we do have some ways of doing that today. On the gene manipulation side, as you mentioned, there's a lot of work being done in that area. And then on the infusion back into the patients, there's two pieces to that. One is the actual putting it into the patient. And we are very blessed in this field that these hematopoietic stem cells know how to get from the blood into the bone marrow. And so that's one of the easiest parts of this process. We literally infuse them the same way that we infuse a red blood cell transfusion. But in order to get them to engraft, we do have to prepare patients. And so today that is done with chemotherapy, which again is very damaging to patients and causes a lot of toxicity and complexities associated with these processes. And so the agents that we've been developing for improved conditioning can both be used in the improved treatment with allogeneic grafts, but also for autologous gene-modified grafts. And we're seeing a lot of interest from the gene therapy and gene editing community amongst use of these types of agents. And you see that through some of the partnerships that Magenta has established with both Beam and Avro as an example. And there have been lots of folks that have been approaching us in similar veins because there is a tremendous opportunity to then make these types of gene therapy and gene editing strategies much safer by combining them with non-genotoxic antibody-based conditioning, like the MGTA 117 agent.
So, Magenta isn't the only one working on this, and your work has been featured in many companies. So moving over and shifting focus maybe to Jasper Therapeutics. So can you tell us a bit about how you got involved with Jasper?
Yeah, so Jasper Therapeutics is really based off of some early work that came out of our efforts here at Stanford when I was a graduate student in Professor Irv Weissman's lab. I like to think of it as the generation one of antibody-based conditioning. When we were initially pioneering this concept, we started to work with a proof of concept agent that was an antagonistic anti-CD117 agent and showed that in immunocompromised mouse models, a surrogate agent to Jasper's agent could be effectively used as a safe and non-toxic conditioning strategy. And based upon those positive findings that we had in immunocompromised mouse models, we had a really nice science paper and then got excited about taking that strategy forward into patients. Because the antagonistic mouse antibody worked in certain settings and given the high needs in this area and in these patients, we wanted to find a surrogate that could work the same way and then actually identified that Amgen had a similar agent against human CD117. And so as a graduate student with my PhD advisor, Irv Weissman, we actually contacted Amgen and asked them if we could get access to this agent that we found out that they had developed for a completely different indication and established a collaboration with them and then brought in a Stanford colleague, Judy Shizuru, to help advance that project forward. And so that's really where the foundation behind Jasper came from. Stanford had then gotten a grant from the California Institute of Regenerative Medicine to push that agent forward. And so we took that Amgen agent and launched a trial in patients with severe combined immune deficiency that is excitingly showing beautiful safety data and early efficacy data. And then the group around Jasper took that agent out of Amgen and licensed it and is now advancing it as a renamed JSP 191 agent from the original AMG 191 agent. Again, I think that there are lots of opportunities in this field for making an impact and for changing the way that we do our current care. And I do think that the JSP 191 agent has some activity, especially in the immunocompromised setting. Our group here at Stanford is also running an investigator-sponsored trial in Fanconi anemia, which is, again, the disease that we talked about earlier, where patients have extreme sensitivities to DNA-damaging agents. And so there are opportunities to use this agent. And I do think that in certain settings, it will show some efficacy based upon our preclinical data. But it really isn't a very powerful approach. And when, again, we've compared the different approaches head-to-head in mouse models, the antibody-drug conjugate is far superior. And Jasper just released some data at the TCT conference testing their agent in patients with MDS and AML. And unfortunately, there were several patients that had relapses, and they have not fully disclosed if the agent itself has any activity in depleting the actual MDS or AML cells. They said that they had garnered that data, but they haven't looked at it yet and haven't reported it, which makes me a little bit nervous. But again, I think that there are a lot of patients who need improved therapies. There's an opportunity for it to be effective, but we're also glad that there's other approaches that are being developed that might be even more powerful, as this initial agent was really taken from a completely different indication and wasn't optimized for this use. But it's amazing that in certain settings, we're starting to see it still have some activity and some utility.
And in that data that you mentioned, I was actually just looking at it over, and one patient they mentioned had late onset grade 3/4 acute GVHD. Did that concern you, or was it only sort of the uptake in inactivity that was concerning?
Yeah, so I think the graft versus host disease is more caused from the inflammatory agents that are still being given to these patients. And in this trial specifically, they're still giving patients total body radiation. And in fact, they increased it in these patients. So initially they were giving 200 gray, now they're giving 300. And they're also giving fludarabine. And the combination of that inflammation that is caused by those agents, plus these allogeneic grafts, I think will cause some graft versus host disease. And so it doesn't totally surprise me. I think we need to eliminate the use of those agents by using more powerful agents that can be used as standalone agents or with other non-genotoxic agents. That's where we'll really see profound changes in the rates of graft versus host disease. We had a beautiful paper a few years ago in mouse models showing that when we gave our antibody drug conjugates in combination with antibody-based immune suppression, we could get completely mismatched grafts to engraft with stable long-term chimerism without any graft versus host disease. And that's where we hope to see the field move to, is being able to do these types of transplant procedures without causing any inflammation, not using any genotoxic agents. And then I think we'll dramatically see decreases in the graft versus host disease as well. But I think, again, the bigger problem with the Jasper trial is that there were some patients that had relapse. They did have the one patient with unfortunate death, bad outcome. And then also just the fact that it is still a suboptimal regimen that does have some genotoxicity concerns.
So moving on, because there's so many companies we can talk about, but let's move on to 47. That was acquired by Gilead in 2020 for about $4.9 billion. It's an immuno-oncology company, focuses on novel checkpoint therapies for cancer, especially targeting that CD47 pathway, and it wants to engage macrophages. So obviously you were involved in the company. I think it was the patent maybe for 47, but would love to hear a little bit more about your involvement and why macrophages maybe, and specifically CD47, were really interesting for you.
Yeah, so I wasn't as involved in the 47 effort. They actually spun that off when I was in Boston completing my clinical training out there. But 47 did license one of my graduate school patents, and then similarly is pursuing a 117 program that they had disclosed. The company, as you mentioned, was acquired by Gilead in 2020 in the middle of the pandemic, and I don't have great insight on what they're doing now. I'm sure they were very busy with lots of different things, but it was wonderful to see that they similarly have been passionate about this area and seeing advances on the conditioning front. And so now we have many different efforts that are working on improved non-genotoxic conditioning, really trying to do targeted approaches against hematopoietic stem cells rather than these broadly ablative approaches. And I think we have multiple shots on goal here that will ultimately transform this field. And so it's been fun and exciting to see these initial concepts and initial studies that work so well in mice really move forward in profound ways now with clinical data in patients and many agents approaching patients in the near term as well.
Right. So another one, I don't know how you have time to do it, but Third Rock Ventures. So consultant there as well. Third Rock Ventures, for anyone who doesn't know, is a healthcare venture firm. You advise companies, also help in company creation, and you've been involved in some really, really interesting companies. So maybe let's do just kind of like a speed round to discuss some of the highlights there. So firstly, Global Blood Therapeutics. very interesting company focused on hemoglobinopathies and sickle cell, beta thalassemia specifically maybe. So the company said, I thought this was interesting, that they wanted to focus on the indication and then find the right drugs for it rather than the other way around, which is what most companies kind of do. It's an interesting premise. What do you think kind of about that premise from a company formation perspective? And maybe a little bit of context on your involvement in the company would be interesting to hear too.
Yeah, absolutely. So Third Rock has been a fantastic group to work with. I consulted with them for almost 7 years and had a wonderful run working with a lot of great people there on a lot of great companies that have been highly impactful and are doing great things for patients. And I first started working with them actually right after we had done the work with the Amgen agent around the CD117 antibody-based conditioning, where I got disheartened by how slow and inefficient the academic system was to really take drugs and translate them into therapies for patients. I think we're much better now than we were back then, but we really didn't have the infrastructure to do that very effectively. Plus, the incentives weren't aligned, and we didn't have as many people with expertise in drug development on the academic side. And so I wanted to learn those skills and wanted to work with people that had developed them in various different ways, and started to work with Third Rock on understanding how they do company creation and how I could be helpful in those efforts. And GBT was the first effort that I had worked on together with Charles Homsey and Craig Muir, who were passionate about trying to do something different for sickle cell. And specifically, Charles is a physician and a cardiologist and kept relating back to his experience in taking care of these patients, where we really had nothing to offer them but pain medications and IV fluids, and pressured us to think about, could we do something better, and put together a small team of folks to explore the area and think about where there was some innovative science that we could build upon. And it was really remarkable to go from concept of sickle cell is bad, I wonder what we could do, to a drug that has looked so amazing over a very short period of time. And to put things into context, we had started this relationship with Amgen around the AMG 191. That drug had been in patients already for a completely different disease indication, showing safety. And it took, I think, 7 years to go from that to actually launching a transplant study in patients with severe combined immune deficiency. Whereas working with the Third Rock group and at GBT, we went from sickle cell is bad, I wonder what we could do, to having a drug in patients in 4 years. And so I think that really shows that if you put talented and passionate people together with the right resources and the right structures to really incentivize collaboration in profound ways, you can do things incredibly efficiently. And the GBT small molecule, I think, is very powerful at unsickling sickled blood cells. And there is a lot of opportunity to use that for the benefit of patients, which we're now seeing through the clinical data. And although we're really excited about curative approaches for this disease as well, through the various different types of hematopoietic cell manipulation therapies, I do think that there's a huge opportunity to treat these patients prior to those types of agents to really decrease the inflammation in their marrow, decrease their sickling, or for patients who don't want to move forward with transplant procedures, especially today when they do have these genotoxic conditioning agents that lead to infertility and all sorts of other comorbidities, to treat them with the GBT agent, either for life or for an extended period of time prior to improved transplant procedures.
You definitely answered my next question, which was the company has a daily oral for sickle cell. And so, just thinking about a daily oral versus, a gene editing therapy, we talk about a lot for sickle cell. How would the market kind of respond to both of those opportunities? But I think you did a nice job of kind of laying out what kind of the promise could be for that. So that's really helpful. Maybe to go to the next one, Bluebird Bio, which we have heard a lot about. It's been in the media certainly a lot the past year. They're a gene therapy company focused on, at least at first, hemoglobinopathies. Certainly, we've seen them have some major setbacks, unfortunately, with their lentiviral vectors. But maybe it would be helpful just to get some context. So, how are you involved in the company? What are you sort of seeing as being someone who was very involved maybe in the beginning, but maybe not as much now, in terms of their setbacks for their serious adverse events that obviously were connected to the lentiviral vectors?
Yeah, so Bluebird was launched long before I joined the Third Rock team, and so they were already an existing company. Given my background in hematopoietic stem cell therapies and non-genotoxic conditioning, I helped the company in some smaller ways, but have been following them very carefully over the many years. And I think that they have a lot to be proud of. They've really been one of the pioneers in gene therapy and have some beautiful data that they've been able to help a good number of patients. I actually just chaired a session at TCT where there were 2 presentations on the long-term follow-up of patients using the bluebird gene therapy for beta thalassemia, showing beautiful long-term efficacy in those patients and nice safety. I do think that they're getting a little bit of a hit just because there are newer approaches that are in development, but we don't know what the safety and efficacy profiles of those truly will be, whereas bluebird has a long-standing history of treating patients, and we have a much better understanding of what the therapeutic profile of their approach looks like. And I think that there are opportunities for them to continue to work with others in the field to move that therapy forward and to enable its use in many more patients, but to also take all of their learnings and work with others on newer approaches that potentially may be better.
Yeah, I think that's a great summary. Yeah, and we've seen the data, which looks promising, but obviously, the company has had several setbacks, but it doesn't mean that the newer modalities won't have some challenges as well in the future. Speaking of a newer modality, Editas, you were also involved in the company. We've spoken about Editas a lot on this podcast. So it's obviously a gene editing company. They use, CRISPR-Cas9 as well as CRISPR-Cas12a. Their first focus was in LCA10, which is an eye condition which can cause blindness. That's their first clinical indication. Just curious, how did you advise Editas? What was kind of your involvement in the company? Were you part of that founding team, and kind of any updates on what you think of the company to date?
Yeah, so Editas was a super exciting company that really was the first of its kind to go into the gene editing space using the CRISPR-Cas9 system. And I was part of an initial group of folks that were exploring opportunities to put a company together, working with Third Rock and a few of the other investment funds that were excited about the space, and working with a number of the academics and clinicians that had been working in those different types of areas. We were very proud of the team that we put together and the approach to really try to take this new technology and transform the care of patients. Editas, similar to many of these companies, has a long pipeline of different disease indications where they hope to be very impactful. But the LCA12 eye condition, I think, is a particular unique opportunity to use this type of therapy in a way that will be very important for patients that have currently no treatment options available. And so I think that part of the drive for moving this treatment forward in that disease is that otherwise these patients go blind, and could we do something that really would transform their care? And this technology had really that potential. It's been interesting to see other groups going after diseases that have alternative therapies, and it's been surprising just how receptive the community has been to gene editing and how quickly people have used this in so many different contexts to become comfortable with it. I think the prior conversation around bluebird is a very interesting one. We have so many different ways of trying to treat sickle cell anemia. And do you use a CRISPR-Cas9 or do you use a lentivirus as an example? We have a lot more data with lentiviruses. Is it appropriate to try something brand new when there is something that works so well? But the field has seemed to feel really comfortable moving with that, and patients are enrolling on those trials. But for diseases such as LCA12, there really is no alternative, and that's why that company in particular felt that that was such an important disease indication to go after.
And the FDA guidance that came out, I think several weeks ago now, did say that the FDA would prioritize or look favorably on programs that didn't have alternatives. So I think that's a really good point that you brought up, and something the FDA is obviously thinking about as well.
Yeah, I think it's really important that we are developing therapies for patients that really have no options. There's so many grievous conditions out there. And I also understand the need for developing improved therapies for diseases that we do have therapies available for. But it is a trickier conversation. I think we need to be really careful about how we consent patients, how we explain risks and benefits. But for these diseases where there's really nothing, I think there's a particular need to do something powerful. I've also had the great fortune of being the clinical PI on the Rocket Pharma gene therapy trials for Fanconi anemia subtype A. And for those patients prior to developing bone marrow failure, we also have nothing to offer them. And you can see how patients really want to enroll on trials and try new therapies that could transform their future care. And those trials have been super fast to enroll on for that reason. And I'm glad the FDA is following patients in encouraging the development of these types of therapies for these awful indications where nothing is currently available.
So another company that you were involved in, curious to know the exact involvement, whether you were on the founding team, consulted, et cetera, but was Decibel Therapeutics, company which is focusing on hearing and balance disorders. So would be curious to know the affiliation that you had with the company. I'm also really fascinated by this company in general. I don't think we focus enough on how important hearing and balance disorders are. But as you mentioned, sometimes when there are other options, should gene editing or gene therapy be some of the first to consider? So curious on your thoughts about Decibel.
Yeah, absolutely. So Decibel is a very exciting company to work on in an area that was very new for me as well. As you've heard from our prior conversations here, a lot of what I focused on has been genetic diseases, cancer, and thinking about the blood and the immune system. Decibel is really focused on improving therapies for hearing loss, which is an area that I had not previously worked on. But I think that there's a ton of unmet need in that area. And as I started to learn about that, really started to understand even more so just how devastating these diseases are for patients. The lack of hearing or the development of tinnitus can be super disabling. And so we had really worked together with a number of different folks on thinking about how we can affect this in powerful ways, and spent a couple of years learning about the area, talking to a lot of smart academics, putting together a team that is now developing several different therapies focused on improving treatment for patients with hearing and balance disorders. At the time, it was really the first of its kind. Now there's several other companies in this space, and I'm glad the area is getting more attention. But prior to Decibel, mostly we just had hearing aids that were available.
So not sure how you have time, but you're also an advisor to some other companies. One example is Beam Therapeutics. That's a company we've talked about a lot on this podcast, and actually recently did a podcast with John Evans, but obviously the base editing company. So curious to know what kind of capacity you were advising Beam in. And then also in reading Editing Humanity by Kevin Davies, who we've also done a podcast with, your name came up in terms of being the one to name the company. So I would love to hear the story of how you came up with the name Beam for the company as well.
Yeah, so I pride myself on being the godmother of Beam. I feel like I blessed it from a distance. It's been a great company to work with, and I'm very proud of all the work that they're doing. But really, my involvement was more on the initiation of the company and through my collaborations with David Liu, who is one of the founders of the company. I got to know David actually while we were building Editas, and at the time we were working on building out the pipeline and thinking about indications where that modality could be really efficacious. And I kept telling David that we have to be able to fix mutations. It's not just enough to be able to cut out portions of genes. And a couple of years later, David came back and said, I think we have something that could do that. And so we got really excited about exploring that further and thinking about how that could be translated into treatments for patients. And as we were thinking about creating a company in this space, David asked me if I had any ideas for a company name, and Beam immediately came to me. Because rather than the original CRISPR-Cas9 technology that is more like a scissors that causes cutting, the Beam technology is more like a laser beam that goes to a specific part of the DNA and allows the exchange of one nucleotide. And then I also liked that Beam stand for base editing. I wanted the A and M to be advancing medicine. I think David ultimately said that for him it stood for and more. But either way, I think it's a great name for an incredible company that's trying to further develop important translational therapies for various different types of diseases.
That's great. I did not know about what Beam stood for. So that's really interesting. So thanks for sharing that. Also, moving on to another company, Spotlight Therapeutics. So can you tell us a little bit about your role there? Were you an advisor? The company, just for some context for anyone who's listening, is focused on in vivo gene editing. They developed a new class of biologics, which are engineered ribonucleoproteins. The company has called this TAGE. and that stands for Targeted Active Gene Editors. So can you explain why this could be potentially helpful, and maybe why current gene editing technologies can't be addressed in vivo without it?
Yeah, so being a physician, I think I just see a lot of medical problems, and that's what really inspires me to try to help various different types of efforts that are trying to address those problems, and why I've been involved in a number of different companies as an advisor. With various different types of genetic diseases, it is wonderful that we have the opportunity in certain cases to be able to take out their cells, fix them, and give them back through these ex vivo manipulation techniques that we spoke about. But it would be even easier if we could just administer these therapies directly to patients. And Spotlight in particular is working on developing an in vivo gene editing technology that would enable delivery of these types of gene modification techniques directly into patients, which could potentially enable expanded access and easier use of these types of therapies. And so I've just been doing a little bit of advising, helping that group as they continue to progress along. So, lots of opportunities to help lots of different efforts that are trying to approach these diseases in various different ways, and just trying to use a little bit of my time and knowledge and expertise to help them along, to hopefully develop these therapies more efficiently, so that we can have them in our toolbox of treatments to be able to give to patients to really transform the course of their lives in powerful ways. Because again, we're very grateful that we have what we have as therapies for patients, but there's a lot that we can do to really improve upon them. I'm very proud of the companies that I've worked with to date that are trying to transform the field in various different ways, and grateful to the broader community for the support of these types of companies that I think is critically important to garner the resources to then be able to move these types of approaches forward and really turn them into approved therapies, and that we have the opportunity to then administer more widely to our patients outside of just the clinical trials that many of them are being tested in today.
So we talked about, other possibilities. So what other areas of medicine interest you, and where else do you maybe see opportunities for innovation?
Yeah, so I think that there's lots of different problems in science and medicine, and it's wonderful to see people doing work in various different areas. But one area that I've seen a lot of under innovation in is the women's health area, and I've been spending some time exploring that space, trying to encourage and support entrepreneurs in those areas, because I would love for us to be able to treat some of these women's health conditions in the same way that we're treating some of these other hematologic diseases, to really have more powerful targeted approaches that can transform the treatment of these diseases. And so I highly encourage the broader community to learn more about some of these disease indications, and to think about how we can be helping with the treatment of women in various different subpopulations that don't have the same types of therapies available today as we see in other areas.
Yeah, so you just mentioned maybe an example where you were potentially helping a company or young entrepreneurs focus on women's health. But, I've read a lot about the fact that you help and mentor young scientists, and especially those with kind of interesting career paths. So I would be curious to know any stories you can share about that, or maybe even any advice that you have for some young scientists in the field that may be listening.
Yeah, so I think I have had the wonderful privilege of being involved in many different aspects of taking science and translating into medicines. I don't think that everyone needs to be involved in every aspect, but showcasing different opportunities has been important to me in staying on the academic side. To be able to help support trainees at various different stages of their training has been a true privilege and joy. And so we have lots of folks that we help here, people that are in our research group, people that are part of our different types of training programs, and Stanford's just a wonderful place to both inspire various different types of trainees, but then also enable them to do incredible things. And I've had personally, the great fortune of taking a concept around non-genotoxic conditioning, proving it in animal models, and now turning it into various different types of therapies that are starting to transform the lives of patients, and hope to get a chance to do that through more of our efforts, but also inspire students and early scientists at different stages of their training to think about where there are problems, where there are things that they're passionate about, how to work with people in collaborative ways to really take those early findings and translate them into therapies for patients. And so lots of different places, again, that people can be involved in that journey and just helping them find those opportunities and encouraging them to really pursue them. As I think we need more and more people in this field that are passionate about it, that are smart, that are hardworking to really completely transform the way that we treat patients.
Well, I think you are definitely helping with that and will, I'm sure, continue to do. And I'm sure we'll hear more from you in the future. But for now, thank you so much for joining. Really great conversation. And I'm excited for everyone to hear it. So thank you so much.
Thanks again for the opportunity. It's lovely to get a chance to talk about some of our work and looking forward to staying in touch and talking more about it in the future.
ARC believes that the information presented is accurate and was obtained from sources that ARC believes to be reliable. However, ARC does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARC. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARC's current views and assumptions and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.